Wasson, who is our last and final seminar speaker, last but not least, ending on a high note. So Jadiel got her degree from UGA. She was a UGA undergrad, turned professor at NYU. And her lab aims to gain a deeper understanding of the mechanisms involved in cross-generational signaling. So she studies how the perception of different environments drive the mechanisms that govern what information is opted for communication to offspring, using C. elegans as one of her model systems. And she uses a combination of genetics, systems biology, neurobiology, and genomic approaches to understand epigenetic inheritance mechanisms. And that's what she's going to be talking about today. So, anyway.
Thank you for the kind introduction. Thank you for inviting me here. It's really great to be back at my alma mater. So I will say go dogs, because I feel like I have to do that every time I step on campus. So, all right. So like Holly mentioned, my lab is really interested in understanding how different environments can impact what parents are passing on to their offspring. So my journey actually started with thinking about something a little different. Yes, so my journey really began with thinking about what happens between these two different cell types. So when you think about sperm versus egg, these are very distinct cell types that have very distinct transcriptional programs. But at fertilization, they somehow fuse together. It's clicking, it's fine.
Fix this real quick.
All right, that's okay. All right, so when you think about those two different cell types, they're very distinct and separate, but somehow they fuse together to create this one cell type that goes on to give rise to every single cell type in the body. So as a graduate student, I really got interested in understanding how this process happens, right? How do you go from very distinct cells to a cell that's essentially a blank slate? And the answer is in short, is something called epigenetic reprogramming. So in order for the new generation to begin, you need to erase a certain amount of epigenetic information in order to create that blank slate. So as a graduate student, I really got interested in this question with understanding, what needs to be erased between generations. So I worked in mice and I focused on a particular epigenetic modifier that's responsible for erasing epigenetic memory between generations. And some of the things that I found was that when you completely get rid of this enzyme, you get a complete developmental arrest, which tells you how important it is to erase generational information and start off with a clean slate. However, some other things arose when we looked at hypomorphic allele, or an allele of this enzyme that where this enzyme is half functioning, right? So when this enzyme is half functioning in the early embryo, what we see is that some embryos can survive and make it to adulthood, but they have these huge behavioral and structural abnormalities. And one of the interesting points that I want to highlight here is the fact that we get these imprinting defects. So the interesting thing about imprinting genes is that you have this parent of origin specific expression. So depending on if the allele comes from mom versus dad, that's the allele that has to be expressed, right? And what's interesting about about these genes is the fact that their epigenetic state is maintained between generations. So it's really important to have to maintain the epigenetic state at these specific genes between generations. So this is an example of a class of genes where you have epigenetic inheritance, where the state needs to be maintained. So in going from thinking about one side of the equation where you have to erase information between generations, I began to think about the other side of that equation where there's certain types of information that need to be maintained between generations. And one of the things that I began to kind of get more into is, what types of information can actually be inherited, right? So this is one example of inheritance with imprinted genes, but what other types of information can actually be inherited? So in thinking about that, I began to expand my viewpoint from focusing on, oocytes to really thinking about the parent and what the parent is experiencing and how that can influence these mechanisms of epigenetic inheritance. All right. So And expanding that kind of even further, thinking about different environmental inputs that that parent is experiencing and how those environmental inputs and the perception of those are influencing this process. All right. So there have been several studies that really have begun to probe how different environments can impact this process of epigenetic inheritance. But in general, there's still not a lot known. So for example, we know that parents can pass on information in the form of DNA and gene mutations. But when you think about this type of information, it's really slow to adapt, right? So how can this really be beneficial to an organism in the case where it needs to rapidly adapt to a particular environment and inform its offspring about that environment? Okay. All right. So during the time I was a PhD student, there were several studies that really came out looking at how different environmental influences can affect what parents are passing on to their offspring. So when you think about things like stress or things like viruses or starvation, these are true experiences that the parent has, right? So an animal starved is going to respond to that starvation. But one of the things I began to think about during this time is, okay, what if an animal is just perceiving that it's stressed or perceiving that something is a threat? Can that also be interpreted as a stress? All right. So one of the studies that came out during the time when I was a graduate student was a study from the Wrestler lab, where they took mice and they exposed them to a particular scent, right? So the smell of cherry blossoms. Every time they expose these animals to the smell of cherry blossoms, they shock them. So they learn to associate the smell of cherry blossoms with being shocked. So they learn to fear that smell, right? So one of the cool things from the study happened next, where they took the offspring from these male mice that were conditioned to fear the smell of cherry blossoms, and they exposed their progeny to the smell of cherry blossoms. And without being conditioned to fear that smell, they innately feared the smell of cherry blossoms, right? So something that was initially, kind of innocuous, like a pleasant smell like cherry blossoms, was recoded in the parent to be perceived as something as a threat. And this perception was then passed on to the offspring, and the rewiring also occurred in the offspring, where they also had this innate fear of that smell, right? So when you think about this, it's really the environmental cue that's influencing the phenotype of the offspring. So here we still are trying to understand the mechanisms that are in play here that connect the sensory perception of the parent to the response in the offspring. So how exactly is this being communicated between generations? And once again, this is really an incredibly fast adaptation to an environmental cue within the span of a generation. So like I mentioned earlier, genes and DNA mutations are very slow to adapt to environmental cues. But on the flip side of that, like you have that epigenetic inheritance, and this is where my work really lies. Okay. So one of the things my lab is really focused on is really understanding can perception and how perception influences the information that's being inherited between generations. Okay. And we really focus on the maternal aspect of this. So what is being maternally deposited into the embryo to communicate this perception to the offspring? All right. So today in this talk, what I'm going to be doing is addressing three major questions that lie in this realm. So one, what are the environmental signals that are perceived by parents, and how exactly are they perceiving those cues? What are the networks, the sensory networks in play that allow the parent to sense those cues initially? And then what is the nature of the information that they're passing on to their offspring in response to those cues? And then ultimately, how is the offspring interpreting the cues that they're receiving from their parent? Right? You can interrupt me at any time to ask me questions. Feel free. All right. So to answer these questions, so essentially I'm trying to understand how you get from that parental generation all the way to the offspring, F1. So connecting the dots between the two. All right, to address these questions, I've turned to using C. elegans as a model organism. And there's a variety of reasons why C. elegans is a great model system for studying inheritance mechanisms. So one, they have these incredibly fast generation times. And they're really responsive to different environmental perturbations. So you can treat them with toxins, you can change the oxygen concentration in the environment, and you can look at readouts in their behavior and other molecular readouts to see how they're responding to those different environmental perturbations. Two, we have recently mapped out their entire connectome. It's not so recent anymore, but more and more recently, we've mapped out their entire neuropeptidome. So what that means is that we know what neurons are talking to each other at the synaptic level, so how they're communicating at that hardwired level. But we're beginning to understand how they're talking to each other at that wireless level, where the neuropeptides are communicating with a mode of communication between these different neurons. They also have very easy genetics, and they're very amenable to biochemical approaches, and also amenable to drug screens. They're a species. They have this deterministic development, and I recently updated this slide to highlight that there are 8 Nobel Prize winners that did their seminal work in C. elegans. Okay, so it's a very powerful model organism. And lastly, they have this incredibly well-defined toolkit, right? And to answer the first question, or this last question that I posed here, how is the information being interpreted by the progeny? I have utilized one of those tools in that toolkit that we have in C. elegans. And that is something called RNA interference, or RNAi. So RNA interference is what happened. It's essentially when you expose the animal to a double-stranded RNA. And you can think of it as this like really ancestral immune response that an animal has, right? So when it's exposed to a double-stranded RNA, it mounts a response that results in targeted gene silencing in response to that double-stranded RNA. So we can feed it a double-stranded RNA to gene X and then look out the silencing of gene X in response to that treatment, right? But this pathway has many ties to other biological pathways in the worm, including looking at translational output and development. So looking at the RNAi response in these animals is really powerful to understand, how they're integrating different signals at the level. So one of the things that we turn to use is looking at RNAi sensitivity in the offspring, and using this as a proxy for the stress response to a sensitized protein. Right? So what does that look like? So you can think of different environmental inputs that a worm is exposed to as informing the parent as to the quality of the environment. And it can then communicate that information to its offspring, and the offspring can mount a particular response. So the parents perceive that they're in a good environment, they can communicate this to their offspring, and the offspring can have, say, a more like suppressed stress response, versus if the parent feels like it's in a bad environment, then the progeny can, they communicate that to their offspring, therefore their offspring have a more mounted response or an enhanced response to that. So the way we use our RNAi assay is that we take if we expose parents to a particular double-stranded RNA, and we look across a range of different double-stranded RNA concentrations, we can actually titrate the response that we see in the offspring. So to really kind of clarify this, we're feeding the parents a double-stranded RNA and then looking at the response in the offspring. And in this particular case, we're targeting a gene that is essential for development. So one of the readouts that we can look at is how well the progeny are surviving, right? So depending on the exposure of the parent, we can see what's going on in the offspring. So if we have a really high double-stranded RNA concentration, the offspring don't survive well, versus a very low, they survive quite well. So using this assay, we choose one particular concentration, so something like a 2% dose, right? It's almost like getting a vaccine, so a mild kind of inoculation. And in a wild type context, we see that animals survive quite well to such a low dose of RNAi. However, if we expose the animals to a certain condition or a mutation under those same concentrations, and we see that the offspring are in fact surviving less well compared to their wild type controls, we can say that one of the ways we can interpret this is that in a wild type condition, these animals are perceiving that they're in a good environment, there's nothing wrong. This is a weak kind of like immune response, it's a vaccine, we're fine. But if the animal is in a particular condition or has a particular mutation where it perceives that it's in a bad environment, they communicate this to their offspring, and the offspring mounts a stronger response to that double-stranded RNA and survive less well. Okay, so this is I'm going to be talking about this assay quite a bit in this talk. So if you have any questions, please ask me. All right. Okay. So one of the ways this looks like at the molecular level is that in those wild type embryos, so we're looking at C. elegans embryos here, where we're looking at the protein product of the gene that's targeted by that double-stranded RNA. And you can see that the protein product is robustly expressed. And these animals will go on to survive. Versus if we have a different mutation, and this is an animal that's mutant, where they're exposed to that same double-stranded RNA concentration, we see that that protein product is not made, and these embryos will eventually die. All right. So this is one of the phenotypes that we're observing, right? So they have an enhanced response to that double-stranded RNA trigger. All right. Okay. So to answer this last question, one of the things we're looking at is the modulation of the stress responses in the offspring, right? And we're using that RNAi assay to proxy them. So to flip to the other side, what are the different environmental signals that are perceived by parents that can actually modulate this response in the offspring? One of the things I really began to think about when I started my postdoc is, what are these different environmental inputs and what's known about something that can be communicated from parent to offspring? And in looking at the literature, a lot of these factors actually have a direct physiological effect on the organism, right? So when you think about something like UV stress, for example, you expose an animal to UV stress, it causes double-stranded DNA breaks. So this is having a direct impact on the offspring, or exposing them to something like an endocrine disruptor, right? This is having a direct effect on, it's a direct genotoxic effect. So what I wanted to know is whether or not I can use something that won't have a direct physiological effect. So I turned to social stress to see whether or not I can, one, determine if it's perceived as a stress, and two, see if I can see a readout in the offspring. Okay, so what does that look like in C. elegans? So C. elegans live in these incredibly social environments where they're constantly interacting with their conspecifics. So they're used to being surrounded by other worms. So I wanted to know what happens when you expose an animal or put it by itself in social isolation, where you don't really have a direct physiological effect on the animal. It's completely by itself. That's the only thing that's wrong with it. Is this something that's perceived as a stress? And is the stress communicated to its offspring? All right, so the way I did this assay is 3 different ways. So one, I had the animals in their normal environment where they're in this group setting. And then at some point in their development, I moved them to a treatment plate to expose them to that weak double-stranded RNA trigger. I allowed them to lay eggs, and then I moved those eggs to untreated plates, allowed them to hatch, and then assayed how well those offspring are surviving. So very straightforward, very simple, right? In an acute social isolation stress, what I did is take those animals, and then at the same point in their development, I moved them to the treatment plates by themselves. So they're just isolated on the treatment plates. They laid eggs in that amount of time, moved those eggs to untreated plates, and assayed how well those progeny survive. And in this last iteration of this experiment, I chronically isolated these animals for multiple generations, right? So from the time they're hatching, so worms lay eggs because they're hermaphroditic, they can self-fertilize. Every time an animal is hatched, it's by itself, it begins to lay eggs, and those first eggs I move. So for multiple generations, these animals have only known that they're by themselves, right? So there's a compounding effect. Then I assayed those animals and offspring to see how well they survive, okay? Okay, so in this particular graph, the way I'm representing it here is that our group controls are, everything is relative to our group controls where it's set to 1. And we're looking at everything as a ratio of that group survival, okay? So in our acute isolation stress, what happens when an animal is just put on a plate by itself for some point of its development, we see that their offspring actually have this enhanced response to that double-stranded RNA trigger. So just by being by themselves for like a period of their development, they're already like, oh, this is kind of stressful. And they're telling their offspring like, hey, there's something going on out here. So those offspring have this enhanced response to that trigger. When we do a chronic situation, what we're seeing is an even more dramatic effect. So when an animal is kept in isolation for multiple generations, their offspring survive even less well to that same concentration of double-stranded RNA, all right? So we're seeing this compounding effect across generations, all right? So one of the things that this told me is that social isolation is indeed a strong stressor in C. elegans, and it's something that they can communicate to their offspring, all right? So in this case, these worms are completely by themselves. They're not touching any other worms. They're not sensing any other worms. I wanted to create a situation that's kind of like an empty club. So imagine that you go to a club the night after like a huge party, like the night after Halloween, right? You go in there, you can smell, you can taste that there were people in there partying and hanging out, but you're like still by yourself, right? But you know there were people once there. So I wanted to create a similar situation with these worms, right? So the way I did that was I took a lot of worms, allowed them to crawl around the plates where the worms are housed. Then I removed them from those plates and then put the socially isolated worms on those plates that worms were once on, okay? So when I do this experiment, what I see is that in those socially exposed plates, we see that we can rescue the response of the offspring almost back to wild-type levels. So what this tells us is that it's truly the perception of the social environment itself that's being communicated to the offspring, right? So just by exposing them to like whatever these worms are throwing out there, that sweat and musk in the environment, these animals are sensing and it's telling them, you're not alone, things are okay. And they're telling that to their offspring. But this really begs the question as to like, you know, what exactly is being expressed, sensed in that environment, okay? So one of the things that worms can do is that they can actually smell and taste their environments, like most organisms do. And one of the things that they're smelling and tasting in their environments are these things called pheromones. And they're really this indicator of their social environment, okay? So when we go on to look at the enzymes that are responsible for producing these pheromones in C. elegans, and we knock them out and we look at the different mutants, the way I'm representing the data here is slightly different, where we have these paired experiments where we have our wild type, we're assaying our wild type offspring compared to our mutant offspring. So if points lie along this dotted line, that means that mutant and wild type offspring are surviving at the same rates. So there's no difference in like that mutant genotype and its effect on its offspring, right? However, if we see points that lie down and away from this line, kind of like what we saw with the isolation, that indicates a more enhanced response in the offspring. Okay, so when we knock out different components of the biosynthetic pathway for the production of these pheromones, so these animals aren't secreting those social cues, what we see is that across the board, we get this enhanced response in the offspring, okay? So it's really about those pheromones that they're sensing. And then much like doing that empty club kind of scenario with these worms, we can expose these animals to crude pheromone from other animals, and we can rescue that response back to wild-type levels. Okay, so it's really about the perception of these social cues that these worms are responding. And one of the things about these pheromones is that they're composed of these molecules called ascaricides, and ascaricides are a large class of these modular molecules that are made up of different. Their building blocks are composed of different components of different metabolic processes in the animal. So really breaking down which components contribute to this intergenerational signaling is really the key to understanding what are the specifics of the environment that the animal is sensing to communicate to its progeny. So these molecules, which are a class of hundreds of molecules, they actually tell the worm not just about its social environment, but also about other things like whether or not it should avoid something, whether or not it should go down a different developmental pathway, tells it about, you know, whatever mates are in the environment. So they're really complex, molecules. So currently in the lab, there's a technician, Dariana, that has been delving into what are the specifics of these cues. So she's been looking at different receptor mutants to see which ones recapitulate this enhanced response that we see in offspring in order to really zero in on the specific ascaricide molecules that are responsible for triggering this response in the offspring. Okay. All right. So the question, of course, is, you know, why does an animal really care about being by itself, right? What is the effect of social isolation? So there have been studies across a number of model organisms that really highlight the impact of social isolation on an organism itself. So in mice, you can see that social isolation actually leads to a reduction in the dendritic spine volume and a change in the synaptic plasticity of the neurons. In flies, when you put flies by themselves, they actually sleep less and eat more. And in another nematode, we see that isolation can actually trigger a change in the completely can change the developmental trajectory of these animals and induce an alternative morph that tends to be more carnivorous versus just eating bacteria, right? So isolation can really like change quite a bit for an organism. And also in bees, we see that isolation actually changes, destabilizes their breakdown. Now, C. elegans, there hasn't really been a lot to probe what does the social environment actually do to C. elegans? But one of the things that we do see is that there's a difference in its germ cell development. And we see that there is a change in the amount of communication between somatic cells and the germline when in these animals that have been isolated. So in this case, we're looking at extracellular vesicles. We see that there are a lot more when an animal is in isolation compared to an animal that's grouped. And lastly, one study that came out recently that really got us thinking about the impact of social isolation on an organism, how that impacts its sensory perception, is that when animals are by themselves, they actually don't respond as well to environmental stimuli like touch. And this is that also impacts how their neurons are developing and how they're structured. Okay, so one of the things that we're beginning to think about is that, how that experience of social isolation is actually changing the brain structure of these animals. And that's one of the things contributing to what they're passing on to their offspring. Okay, so to answer this first question, what are these environmental signals that are perceived by the parents that can be communicated to the offspring to modulate these responses? And I just told you that there's social cues that are perceived by the parents that affect what they're passed on to the offspring. All right, so the next obvious question, of course, is like, how exactly are they sensing these cues? So like I said, they can smell and taste their environments. And one of the ways they do that is through these chemosensory neurons. So the cool things about these neurons is that they have these ciliary endings that are exposed directly to the environment. And at the end of these cilia are a variety of receptors that can respond to different chemicals and cues out there. So they're directly, they're responsible for the animal's perception of their environment. And in C. elegans, like I mentioned before, we have the whole connectome mapped out. We can genetically ablate specific neurons and ask how, when you get rid of that particular neuron, how does that affect a particular readout? In our case, our RNAi response in offspring. Here, I'm just showing you this really cool tool built by someone in the Hobart lab, where they have basically expressed the fluorophore in every single neuron, all 302 neurons in the worm, so that when we image these animals, we can say that, okay, we know that this neuron has this particular identity, AIN, so that when we genetically ablate it by, expressing a caspase in it, so it's, it undergoes apoptosis, we can say when we don't have AIN, what happens to the organism, right? So there are a lot of really cool, powerful tools in the worm to get at sensory perception and general effects on behavior and animal physiology. So here, when we went on to assay what happens when there are different chemosensory neurons that are ablated in these animals, I'm showing you this data once again in this way, where you have wild type. If mutants survive the same rate as wild type, they don't have a change in their response. However, if they're points above this line, that means that their offspring have a suppressed stress response. And then once again, if the points are down and away from that line, it means that they have a more enhanced stress response, okay? So we're looking at survival, and that's indicative of the offspring's response. Okay, so when I went on and assayed about 8 different neuron pairs in these animals, when you genetically ablate these neurons, so these neurons are never there, these animals survive, their offspring survive at the same rates as wild type, so there's no effect, right? However, when I went on and assayed 5 different neuron pairs, 5 other neuron pairs, what I saw was that we had these suppressed responses in the offspring. So by getting rid of these neurons, somehow this tells the parent that, hey, things are really great in the environment. It tells that to his offspring, and their offspring are like, hey, I have a suppressed response. I can handle anything that's being thrown at me. Suppressed stress response. Then when I ablate 3 other neuron pairs, I see that those offspring actually have these enhanced responses to the stress. So we're kind of titrating these responses between suppressed. All right, and I'm just going to zero in on one specific neuron pair, the ASI neuron pair, because we know the name of the neuron in C. elegans. And when we get genetically ablate that neuron pair, we see that the offspring of those animals have this enhanced response. Okay. All right. So one of the things I would like to highlight is that one of the things we're thinking about is that at the sensory level, at the circuit level, we're beginning to think that there might be an emergent circuit for the regulation of intergenerational signaling, right? So I've color-coded these neurons where the ones in green, get rid of them, their offspring have an enhanced response versus the ones in purple, the offspring have like a suppressed response. And we're just wondering, this is what I'm highlighting here is the known connectome of how these neurons are talking to each other at the synaptic level. So there's a lot of like communication between these neurons. So one of the questions that my lab has is whether or not we are identifying a particular like sensory circuit that's responsible for triggering epigenetic inheritance or intergenerational signaling dynamics. And, you know, to make this an even more complicated picture, a lot of these neurons have been characterized in terms of like what their role is in the animal. So some of these have a role in chemotaxis, social behavior, immunity, behavioral plasticity, sensing temperature. So there are a lot of different environmental cues that are being integrated in these neurons. And another question that we're trying to understand is like beyond social cues, you know, what are other sensory inputs that can modulate this circuit in order to tune how the parent is passing on offspring to information to its offspring. Okay. All right. So to continue to walk down this path, I highlighted that ASI neuron pair. And one of the ways, you know, one of the questions is how do you get from that neurocircuit level to like offspring, right? We have to somehow communicate to the germline. So one of the ways that neurons communicate with each other is through neuropeptides. So we went on to assay all 3 major neuropeptide families in C. elegans. So we assayed about 17 different neuropeptides. And what we saw was that for the large part, a lot of them did not elicit any kind of change in the response in the offspring's response. However, I'm going to highlight one particular neuropeptide called FLIP21, which happens to be an MPY homolog. We have MPY, it regulates a lot of functions. When you get rid of that neuropeptide in C. elegans, the offspring have this incredibly enhanced response to the parental exposure to double-stranded RNA. We can rescue that response back to wild-type levels by reintroducing a wild-type copy of that gene into these animals. And then we can also overexpress this gene in C. elegans. And what we see is that we get a more suppressed response in the parent. So what this is telling us is that the levels of signaling of this particular neuropeptide pathway can actually tune the offspring's response to a particular stressor, right? So it's really the amount of signaling that's dictating the offspring's response. All right, so I mentioned that this is an MPY homolog. It's orthologous to MPY. And what we see is that, I'm gonna speed up here, is that some of the same functions that this neuropeptide is regulating in C. elegans, it's also regulating in mammals, like metabolic functions, stress modulation, sleep. And these neuropeptides in C. elegans, which are highlighted here, and I have FLIP21 here, you can see that it can actually activate a lot of the MPY receptors in humans. So they're quite functionally similar, right? Okay. So in order for these neuropeptides to act, they have to, of course, bind to GPCRs or receptors. So we went on and assayed a number of FLIP21's receptors. It's known that it binds to 9 different receptors. We assayed 7 of them. For the most part, when you get rid of these neuropeptide receptors, we see that they phenocopy what we see with FLIP21. And I'm just going to highlight one in particular, MPR11, that has this really strong enhanced response in the offspring. So I highlighted these few specific pieces because one of the things we've done is epistasis analysis to show that these components are actually acting in the same genetic pathway to regulate the stress response in the offspring. Right, and so we have social cues in the environment this ASI neuron pair, the FLIP21 neuropeptides binding to that NPR11. And we see that together, all of these components are acting in the parent in order to modulate the offspring's response. Okay. And one of the things that we're currently trying to figure out, if I show you that same diagram I highlighted before, where we know where FLIP21 is expressed. Its receptors are actually expressed across many of these different neurons. We know that the receptors are also expressed in other tissues. So this really creates this complex picture of like, okay, where is FLIP21 coming from and where is it signaling to, right? And that's one of the things that we're trying to figure out now, where there's a graduate student in the lab that's trying to tackle this question, where if we can rescue NPR11, for example, in a specific neuron, can we rescue the phenotype in the offspring? So we can begin to tease apart, track from neuron to some other tissue, all the way to the germline to see where the signaling pathway is signaling to in order to modulate what's happening in the offspring. Okay. How am I doing on time, actually?
Well, 10.
Till 10, okay, what time?
Like 10 more minutes.
Oh, right, you run. Okay. So of course I told you all this about, this being a intergenerational signaling thing. So, what parent is actually important for this response. And we know that parental effects can be both from the mother and the father. And I always draw the oocyte as being this outsized thing because, well, it's more important for development in my opinion. But we did an experiment to really try and dissect which parent is responsible for passing on this phenotype. And in this particular case, oh man, I forgot to go that slide. We see that in a wild type case where both the offspring are genetically wild type, they have a wild type phenotype. When both parents are mutant, we know that the offspring are also mutant for that neuropeptide, and their phenotype is that enhanced stress response. So the question becomes, what happens when either the father is mutant or the mother is weak? And the offspring are heterozygous or genetically wild type, what is the phenotype of those offspring? So when we do this experiment, we see that the phenotype, that enhanced phenotype that we see tracks with the maternal genotype here. Okay, so the mother has to be mutant for this pathway to trigger this enhanced response in the offspring. Okay, so it's really about the mother's genotype. Okay, all right. So getting to this big question, what is the nature of the information that's actually being passed from parent to offspring in this case, or mother to offspring? We were thinking about a lot of different mechanisms in these animals, and one of the things we settled on is maternal provisioning. So across the animal kingdom, mothers are donating a variety of cytoplasmic components to their offspring in order to help them jumpstart their development. So they're contributing things like proteins, metabolites, RNAs, small RNAs, in order to help it's like a care package to help the offspring jumpstart their development, like I said. So one of the things we did was look at, okay, is there a difference in the level of maternal provisioning in our different animals? So maternal provisioning in C. elegans is driven by two different factors. One of them is here I'm showing you the C. elegans germline. So we have the mitotic germline stem cell pool. And then as the germline continues, these cells differentiate and become oocytes. And then these oocytes can get fertilized by the sperm that's housed here and then become embryos. And one of the things that really drives maternal provisioning is that they can get a lot of components from cytoplasmic streaming that's happening here. So that'll dictate the size of this last oocyte here. However, there's a lot of maternal factors that get pushed into the oocyte that come from other tissues like the intestine. So because these are egg-laying organisms, they actually package a lot of yolk into their offspring. So a lot of the yolk and other components actually come from the intestine and get deposited into the oocyte. So by looking at the size of this last oocyte compared to the first oocyte, we can look at the difference between the components that are donated by the germline versus those that are coming from the soma. So this is really small text here, and I apologize for that. But on this side, we have, we're looking at the first oocyte, or this oocyte, this last oocyte here. So the materials that are coming from the cytoplasm, and we can see that here in a wild-type organism, first to last, we have this kind of like slope here, where there's slow growth in the oocyte so we get basically some cytoplasmic components coming into the oocyte in a wild-type case. But then when we look at our FLIP21 mutants, what we see is that last oocyte is much smaller compared to wild-type animals. So it's getting less components from the germline itself. But when we look at this last oocyte that gets a lot of its factors from the soma or from the intestine, we see that oocyte is much larger. So what this is telling us that by disrupting chemosensory signaling, by getting rid of this neuropeptide, we're altering the amount of like somatic factors that are being packaged into the oocyte that can be passed on to the next generation. So this gave us some hope in trying to understand, like, okay, we know that we have a maternal provision issue. You know, what else is maternally provided by these animals? All right, so one of the things that we decided to do is do some very early RNA sequencing of early embryos to see how, like, what are the maternal RNAs that are being deposited into the embryos themselves, okay? And when we compare wild type to FLIP21 animals, we see like a huge deregulation of transcripts in terms of what's being maternally passed on. And one of the things, we saw a significant amount of changes, but one of the things that really struck us was the fact that a lot of the changes were associated with translational regulation. And the reason why this really stood out to us is because translational downregulation is actually a hallmark of stress responses to many different cellular triggers. And it was really like indicative of maybe there's some kind of integrated stress response that's happening in our chemosensory mutants that then is being passed on to the offspring in the form of these transcripts. So just to like highlight what the integrated stress response is, basically when an animal is exposed to any kind of stress, you get this translational reprogramming where most translational is shut down and you get selective translation of certain transcripts in order to respond to that particular type of stress. And in order to really probe whether or not our animals had, we wanted to know whether or not our animals are translationally compromised. So we did a metabolic labeling assay where we fed them a radio-labeled methionine so that any newly synthesized proteins will get radio-labeled because methionine is that first amino acid that gets incorporated. And when we look at animals relative to wild-type incorporation, we see that we have controls that have lower translation rates. Our FLIP21 mutants also have lower translation rates, as well as some of our other chemosensory mutants where we know that their offspring have this enhanced stress response. So we see the signature of reduced translational capacity in these animals. So we wanted to know if reducing translation alone can also impact the offspring stress response. So we assayed a number of different translational components. So when you get rid of them, you see that the offspring also have this enhanced stress response. So that fits with what the data we're seeing. And I'm just highlighting one particular factor, IFF2. And when we look at the molecular signature of that enhanced response, we see that they phenocopy FLIP21, and we've also done epistasis analysis to show that both this particular translation factor and our neuropeptide are in the same genetic pathway to impact this offspring stress response, right? So I've just told you that, you know, lower translation rates impact stress responses and they're acting in the same pathway as our chemosensory mutants. So the question is, can we rescue the response in offspring just by boosting translation? Okay. So the way we did this is that we overexpress the S6 kinase in order to boost translation. And when we do that, we can rescue our levels back to, in a normal translation boosted animal, we have largely wild type / suppressed responses in the offspring. When we get rid of our FLIP21 mutants, we see they have that enhanced response. And then when we boost translation in a FLIP21 mutant background, we can rescue that response almost to wild type levels. So just by boosting translation, we can rescue our offspring's phenotype. And there seems to be a conserved role for this FLIP21 and PY signaling in modulating mTOR activity. So here I'm just showing that in an MPY mutant that you have increased levels of the S6 kinase. All right, so understanding, hurry up. So maternal provision is actually something that is really key to an organism's development and the eventual like phenotype of the offspring. And This is just highlighting a couple of studies that came out of C. elegans where, the provisioning of maternal ribosomes can actually drive development. And in this particular case, when a parent is starved, the rRNA that's deposited into the offspring are actually methylated, and this is leading to altered translation of transcripts in the F1s or the offspring, and this drives altered response to starvation in the offspring. So there's really something about the translational machinery that's deposited from the parent that can actually influence development significantly. So that's something that we're in the process of really trying to understand. I have a technician in the lab that's developing tools so that we can actually probe this from the embryonic side, where we can look at translation rates and translational efficiency in the offspring in response to altered chemosensory signaling. Sorry, so like I mentioned before, we were trying to probe that social stress that didn't have a direct physiological effect on the animal, but we wanted to know whether or not our mutants had other stress, hallmarks of altered stress responses. So we focus on oxidative stress. So we look in wild type animals. If you expose them to hydrogen peroxide, over time, they eventually die. And this is kind of like the rate of lethality over time in a wild type animal. However, in a FLIP21 mutant, we see that they survive much better to this oxidative stress. And when we look at other chemosensory mutants, we see that they also have a more resistance to this oxidative stress. So it seems like the stress state of these animals is a little like elevated, and they're better able to handle stressors that are coming away, right? It's almost like they're boosting their defenses already, and therefore their offspring are therefore also kind of like ready to handle whatever comes at them, right? And another experiment that we did to really probe other stress responses in these organisms is that we looked at kind of their ability to handle mitochondrial stress, because oxidative stress is something that impacts the mitochondria. So we wanted to know if at a basal level they have higher levels of activity of the mitochondrial stress pathways. So the way you can do this in C. elegans is that you use a transcriptional reporter of a stress responsive factor. And when an animal is just like under like normal control conditions, we're looking at the level of induction of GFP expression. So normally when an animal is not exposed to anything, sorry, let's just cut off, it doesn't fluoresce. And in our case here, this is a wild type animal. There's no fluorescence when you're not triggering the stress pathway. However, in our FLIP21 mutant animals, at a basal level, they have a heightened expression of this mitochondrial stress pathway. And then that level increases when we induce mitochondrial stress. And we can do this with drug treatment as well, and we see the same response. Okay, so in our FLIP21 mutants, what we're seeing is that in general, they have these elevated levels of stress response pathways. And this is somehow creating a particular state that the animal is in that allows it to change what it's passing on to its offspring to prepare it better for the future. So right now I have a series of undergrads, there are even more, that are probing different stress response pathways to see, if it's specific to mitochondrial stress, or if it's specific to other types of stressors, or if it's just a general phenotype that we're seeing. And the cool thing about mitochondrial stress is actually that like an organism gets all of its mitochondria from there. And one of the things that I find interesting is that, like I mentioned before, is there a particular state that these animals are in that make them more likely to trigger epigenetic inheritance mechanisms versus others. And that's one of the things we're trying to figure out. Okay, I'm speaking really fast, but I'm going to summarize it really quickly here. So you can imagine, just summarize the data. I showed you that there are a lot of different environmental inputs that these animals are exposed to, including the presence of other worms that are secreting these pheromones in the environment that they then sense partially through these neurons, this ASI neuron pair that signal through FLIP21 neuropeptides, that signal through their receptors. And when all of these signals are intact and present, the animals get this perception that they're in this good environment, that everything is like great, right? So because they feel like they're in a good environment, they're allocating the proper amount of machinery to their offspring, and their offspring, when challenged, can actually mount a proper stress response, right? However, if any component of this pathway is missing, the way we're thinking about this is that these animals actually think that they're in a bad environment. They can't sense what's out there. Things are kind of awry. They boost their own defenses, so they have heightened stress responses. And this means that they're allocating less materials to their offspring. And in turn, those offspring when challenged actually have a more enhanced response to stress. Okay, so it's really about the perception of the environment that is tuning this intergenerational stress response. Okay, so throughout this talk, I have answered these 3 questions. My lab is actually trying to delve more into some of these different aspects of this system, trying to uncover different environments, different parental states, and really trying to understand how the offspring are interpreting what they get from the parent. And in general, just to kind of like zoom back out, to address my original question, can perception really influence epigenetic inheritance mechanisms? This really ties in both like my graduate work and the work I'm doing now is like really trying to understand how is certain information really opted for inheritance itself, right? So the way I like to think about the generational border is like, it's kind of like this dam, right? Where you have some information, for the most part, you're regulating the information that's passing from the parent to the offspring. So some information, most of it is not going to like pass through. You get that reprogramming, it gets erased. However, there's certain pathways in this dam that allow information to pass through, right? You got the fish coming through, you got a little bit of water coming through. And in this way, this is more like epigenetic inheritance, right? So one of the things that I'm really trying to figure out is like, what are these mechanisms in the dam that allow that specific information to pass through? All right, so with that, I would like to thank my lab. They're really awesome people. My previous advisor. So a lot of the work I talked about today is from my postdoc. I love the reagents that I was able to borrow from people, previous funding. And with that, I will take any questions that you have.
Questions from the room first.
Yes.
Great presentation. I wanted to know if there are any changes in the clutch size when you are giving the stress or dsRNA or changing pheromone, because there are organisms which have like lay more or less eggs when they are under stress, and does that affect the survival percentages you are seeing?
Yeah, so we have assayed the number of offspring that each of these mutants gives off, and there's not a significant change between wild type and these mutant animals. However, in the case of a wild type animal that has been in isolation compared to a grouped animal, there is a shift in their reproductive window, and they do seem to be laying a different number of eggs. But we haven't assayed the size of the eggs because that will also be indicative of like how much material they're getting to, right? And that's something that is like on the dockets for us to do.
Yeah, so when talking about epigenetic inheritance, at least for me, it's hard not to see how it's reminiscent of like Lamarckian evolution, which, when you're in high school or undergrad, they're like, well, there's Lamarckian, which is wrong, and Darwinian evolution, which is right. And it's like, this kind of changes that for me a little bit. So this is kind of philosophical, but my question for you is like, if you were to teach an undergraduate class explaining that, how might you reframe or not reframe how it's usually taught?
Yeah, so I would highlight that Darwin was not anti-Lamarckian himself. Like he actually believed a bit in some of the inheritance of like traits, right? So I think that whole narrative has been kind of like conflated. So I would just kind of clarify that for sure. And a lot of what I'm talking about for like, decades, like people have talked about like maternal effects, right? So this is something that's analogous to a maternal effect. Well, it is a maternal effect that we're seeing. So of course, everything that gets packaged in the gamete and what the moms are exposed to, like that's going to influence development. So that's something that's like well known. Yeah. Right. In the case of epigenetic inheritance, where something carries on for multiple generations, one of the things that I would introduce is really thinking about how do you like maintain that information. And when people think about epigenetics, they think more so about changes at the DNA level or at the histone level, right? So those modifications. So how do you get reliable copying of those marks across multiple generations, and at what point do those get erased? So talking more about like the epigenetic mechanisms from that standpoint, but then also talking about how like from an RNA level, how RNA can actually be like amplified, right? And how that can also act as a signal to be inherited for multiple generations. Does that answer your question? Yeah. Yes, sir.
This might be a naive question, but like, have you like tested how many generations this get to be back to normal, or is it like always gonna be like some remnants of the past?
Yeah, that's something that is on, so we have done an experiment in the acute situation where we have reintroduced the animals back into a group setting and we see rescue of that response. So it is quite classic, but we haven't done the experiment where they've been like isolated for generations and then like reintroduce them to see whether or not there's still an impact from them being like multi-generationally isolated. But I do, that is like one of the questions that we have and that we're testing right now.
And do we have any questions on Zoom or any more questions in the room?
We have here Zoom.
Sometimes they would, if you're on Zoom and you have a question, you can unmute.
I also have the mute chat open.
Okay, otherwise, Olivia.
Okay, so I guess my question is, maybe correct me if this is misunderstanding or anything, but it seemed like some of the mutant was sometimes better at responding to stress. So I was kind of wondering, like, what are the downsides of being so good at responding to stress? Like, you know, why is that a mutant state, I guess?
Yeah, I think this actually gets to a conversation I had with someone at a conference where it's like, you know, what does it actually mean to be adaptive? And is that something that we should like, is that language that we should change when we talk about like evolutionary advantage versus not? So a lot of like when you think about epigenetic inheritance, not all of these are going to be adaptive traits. A lot of them could be maladaptive. So if you introduce it, if an organism is born into a bad environment, you know, it passes on that information to its offspring, but then its offspring end up being in a good environment, you know, its state is mismatched to its environment. So that can be maladaptive, right? So I would say that it's not necessarily a good or bad thing. It's just more so about kind of like the state that the organism is in that's like driving kind of these responses. And depending on the environment that they end up in or whatever they get exposed to, that will dictate whether or not it's a good or bad thing.
We have any Zoom chat questions?
No questions in the chat.
All right, last call for Zoom people to unmute. Any more? All right, well, thank you so much for an amazing seminar.
Thank you for inviting me.
You guys know if there's a lunch?